Hi, I'm Miranda Cosgrove, and this is Mission Unstoppable. Coming up, we're squishing strawberries for science.
We're gonna pull out all of the DNA from the strawberry, and we're gonna be able to look at it. Whoa. It's like cotton candy.
And pets aren't just our friends. They're good for our brain. Many surveys show that dog owners are the happiest pet owners. Then, tuck yourself in as I spill the STEM tea on sleep paralysis. Sleep paralysis may sound scary, but the good news is that it only happens to less than 8% of the general population. And Fig goes farming. In space? Wow! Get ready to meet the scientists, inventors, and heroes who help make our world a better place. The future is here. The mission, unstoppable. Strawberries are great. You can put them in a smoothie or on top of some shortcake. But you can also destroy them and harvest their DNA. Let's see what that's like.
All living beings, from plants to pandas to people, are made up of cells. And inside those cells lies DNA, the blueprint for all the proteins cells need to, well, do everything. But what if you could change a cell's blueprint? Could you create the next life-saving drug? Could you even cure cancer? To find out, I'm meeting with Anjali Chadha.
I really wasn't one of those kids who knew exactly what they wanted to do with their lives or what they wanted to do professionally from a really young age. And I still don't think I do. My real passion for life sciences, I think it's really solidified and become my strongest and biggest passion.
Anjali has followed these passions to MIT, where she studies a subfield of bioengineering called synthetic biology. And today, this 19-year-old is going to teach us about the power of DNA with the help of a few strawberries.
We're gonna go through a long process to basically pull out all of the DNA from the strawberry. It's gonna form kind of like a fibrous clump, and we're gonna be able to look at it.
To collect this DNA, we need to break down the strawberries, mechanically and chemically.
Science is not that dissimilar from cooking or baking. A lot of it is kind of like following a recipe.
We cut the stems off, put them in bags, and squish them until there aren't any clumps. Then we take about a half cup of hot water and mix with a half teaspoon of salt. We take 2 teaspoons of the saltwater mixture and add it to our bag. The salt helps remove proteins bound to the DNA, and it neutralizes the DNA molecule's negative charge. This makes the DNA less soluble in the water. After some more smushing, we add a half teaspoon of dish soap. This breaks down the membranes on the outside of the cells, so the DNA inside can get out. Finally, we pour our well-mixed mush through a coffee filter and wait. We give the filter a quick squeeze, take it out, and pour in some isopropyl alcohol. This alcohol is soluble with the water, pulling H2O molecules off the DNA. With no water molecules keeping them apart, the DNA starts sticking together in clumps.
Oh my gosh, it's like a cloud. It's like cotton candy. Can you believe that this much DNA was in one of these little strawberries?
How do we go from clumps of DNA to something we can actually use?
So that's DNA.
That's DNA.
We're looking at DNA.
Yeah, it's DNA. Not actually individual strands at this resolution, but if you want to actually be able to see that sequence, you can run a DNA sequencing protocol. We, within 24 hours, are able to see like what the exact nucleotide sequence of a piece of DNA is.
Nucleotides are the individual molecules that make up DNA strands. There are only 4 types of nucleotides, labeled A, C, G, and T. A gene is a specific nucleotide sequence that codes for a specific protein. Many thousands of genes work together, creating the proteins needed to run our cells. And Anjali studies how to turn these genes on or off through genetic circuits.
What we've now realized is that we can actually turn genes on
by introducing a certain biological molecule, and we can turn genes off when we want to by either removing a biological molecule or adding in a second biological molecule that does a whole different function.
This work isn't just theoretical. The bacteria in this petri dish, it had a gene added from an entirely different species to give it a brilliant purple color. And the insights pulled from working with bacteria can lead to treatments for humans. What if scientists could figure out what switch can be thrown to tell a tumor cell to stop growing? You could literally stop cancer.
It obviously sounds like some of the research that you're doing really could help people if it down the line becomes successful.
It's really easy to stay motivated by a project when you know like what you're doing on a day-to-day basis could have a tangible effect on someone.
I truly had an amazing time just seeing the DNA of strawberries and learning about the potential just like studying DNA has on a much greater scale. I'm probably gonna go get like a strawberry parfait or like smoothie. If you want to join.
Sounds great to me. I'd love to.
In my dressing room between takes, I like to check in with our experts. So I asked Anjali what she's been up to since we last filmed. Check it out.
Hey Miranda. Since I've been on Mission Unstoppable, I've actually graduated from college at MIT and I started a new job in London. I'm working in a lab where I'm studying how proteins develop and evolve into the shapes that they take in the natural world around us, in other organisms, as well as in our own human bodies. I'm really excited to be working here because the head of my lab is actually an amazing and awesome woman in STEM. To all the young girls watching out there, I just want you to remember, you have to keep believing in yourself and you can do anything in the world that you want to as long as you keep loving what you do and learning about what you're curious about. Bye, see you guys later.
Our pets have power over our brains, but it's a good thing and not like a mind control scenario. Dr. Brain has more. Let's be real. We love pets. In the United States right now, more than half of households have at least one pet. And it makes sense. Look at this kitten. Who doesn't want to have this little guy living in their house? And look at this dog. What a good girl. Look at that face. I just want to hug her. Look at this. I'm sure he's nice. Pet owners are not only happier and healthier, but they actually live longer, which is great news because you get to spend more time with your pet. But having a pet can also mean having a healthier brain. Hi, I'm Dr. Brain. Okay, my real name is Crystal Dilworth, but I have a PhD in neuroscience, which is easier to call me Dr. Brain. So, I'm Dr. Brain! There are many reasons that scientists think that our pets are good for our brain. Simple, routine caretaking activities like getting outside and taking your dog for a walk are good for both you and your pet. And sharing these healthy habits may be one reason why depression is less common in dog owners compared to non-pet owners. In fact, many surveys show that dog owners are the happiest pet owners, which might explain why dogs are the world's most popular pet. Don't worry, birds. Maybe you'll be more popular next year. Not to be outdone, cats have their own superpowers when it comes to our well-being. The low rumble of a feline purr has been shown to increase the release of neurochemicals called endorphins that block pain signals and help to reduce stress. And also, they're in all the best viral videos. The act of petting a cat or a dog also helps reduce the stress hormone cortisol and activates the release of other positive brain chemicals like oxytocin that stimulate feelings of safety, trust, and caring. Scientists showed that the brains of pet owners had less signs of physical aging, with some looking up to 15 years younger than the brains of non-pet owners. They were also better at orienting their attention, certain types of memory, and brain processing speed. So pets keep us active and mentally sharp, especially when they learn how to jump on your kitchen counter and are constantly shredding all of your paper towels. So you have to put the paper towels in the cabinet, but then your cat teaches itself how to open your cabinets and then, oh, that's just my cat. Okay. Keep in mind, in order to reap the neurological benefits of pet ownership, we actually have to spend time with our pets. Just owning one isn't enough. Make a new maze for your hamster. Gaze lovingly into the eyes of your dog, and if you're a future herpetologist, feed a lizard or a snake or whatever it is lizard people do. You'll live longer, be healthier, and your brain will thank you. Bye.
Have you ever woken up and can't seem to move? You can't speak. You open your eyes and see dark figures that aren't really there? This bizarre and frightening experience that many people, including myself, have gone through is called sleep paralysis. So what is it? And how can you stop it from happening? Let me help solve this problem with a big serving of stem tea. Sleep paralysis may sound scary, but the good news is that it only happens to less than 8% of the general population. So what if you're part of that 8%? Maybe understanding what it is can help it seem less spooky. Let's start with sleep cycle. When we go to sleep, we go through 4 stages. Stage 1 is basically when you first fall asleep. This usually lasts 1 to 7 minutes, and it's the easiest stage of sleep to be woken up from. Stage 2 is a more subdued sleep. Your temperature drops, your muscles relax, your breathing, heart rate, and even your brain activity slows down. Stage 3 is deep sleep. And studies show that even though your brain activity is reduced, deep sleep contributes to insightful thinking, creativity, and memory. Once I figure out how to do my job during stage 3, it is over for my enemies. Now it's time for the big event, REM sleep. Even though our eyes are closed, you can see them move rapidly, hence REM's full name, rapid eye movement. During REM, brain activity is almost the same as when we're awake. And during this exact time, our bodies go through atonia, which is a temporary paralysis of the muscles, with 2 exceptions, the muscles that control breathing and the eye muscles. I mean, think about it. If your arm and leg muscles weren't turned off, you would be acting out your dreams. When you wake up during a REM cycle, your body's still going through atonia, so you can't move, even though your eyes are open and you're technically awake. Your mind is also still in dream mode, which makes you hallucinate and think that Hat Man in the corner is real. Science calls this intruder hallucinations. And it's not just paralysis of your body. You might also lose the ability to speak. You can also experience chest pressure hallucinations, which cause you to have the feeling of suffocation. But don't worry, it's not a demon suffocating you. It's just your chest muscles paralyzed momentarily. And finally, we have vestibular motor hallucinations, which include feelings of movement or out-of-body sensations, like flying. Which, honestly, this one doesn't sound so bad. I'd try that. The good news is the paralysis doesn't last forever. Even though the exact cause of sleep paralysis is unknown, there have still been many studies that give us a general idea of the causes, which can be lack of sleep, having inconsistent sleep schedules, mental stress, and even sleeping on your back. So the best thing you can do is to practice good sleep hygiene. For instance, exercising during the day, going to bed at the same time every night, making sure your bedroom is quiet, dark, and relaxing, and removing electronic devices like phones and TVs can all help you fall asleep faster. And now that you have the tools to get a good night's sleep, you can rock yourself to bed with the knowledge that sleep paralysis is just our brain being a little too dreamy. And like all nightmares, it can actually hurt you. That was a lot of work. Time for my nap. Old MacDonald had his farm here on Earth, but future MacDonald might just be farming in space. Take a look.
In many ways, life on the International Space Station is a dream. Cutting-edge science, sweeping views of Earth, and you can do that thing where you float around. But one thing that's missing, plants. That's why today we're talking to Lachelle Spencer, a lead horticulturist at NASA's Space Food Crop Production Facility.
At Johnson Space Center, we pretended to go to an asteroid for 30 days. We were completely cut off, and we had the opportunity to grow little plants as part of experiments. And we all learned firsthand how important it is to bring those smells and those textures with us, because everything else is hard and sterile, and these soft colors and textures has a real calming effect.
Today, I'm at the Kennedy Space Center in Florida to learn how horticulturists like Lachelle are sowing the seeds for interplanetary travel.
So what does it take to grow plants in space?
At home, it can be difficult, but it's even more difficult in space.
Yeah.
The way water interacts in space, it can adhere to surfaces. And sometimes if there's too much, it'll completely adhere to roots and drown our plants. We're also trying to figure out the right lighting spectrum. When we get to LED lights, some plants don't necessarily respond to those narrowband wavelengths. We have the capability of mimicking the environmental conditions in a space cabin environment. We can adjust the temperature, the humidity, and the CO2 so that it's just like what the astronauts experience up there.
What are some experiments that you get to do here?
The most interesting experiments is screening lots of different crops.
Okay.
These crops aren't always happy with that environment I mentioned earlier. We had a number of strawberries that we screened. We've done beans and peas, tomatoes and peppers.
Beans, greens, tomatoes, potatoes, lettuce.
I didn't want to do it, right? Beat me to it. We'd like to be able to use those plants to provide vital nutrients that degrade in their packaged food during long missions, possibly help provide a little piece of home and some psychological support.
These massive environmental growth chambers are filled with some of NASA's other experiments, like how different levels of silver biocide affect lettuce. Or this chamber, filled with a bumper crop of microgreens.
So these are only 7 days old. And what's cool about these is we can grow a lot of produce in a small, compact space. And microgreens, depending on the variety, can have up to 9 times the nutritional content as a mature variety.
By harvesting the crops, Lachelle checks to see which plants thrive or struggle under conditions they might experience in a space capsule.
We're normally weighing them and collecting their fresh weight. We'll dry them so that we can collect dry weights. We'll also send them to labs to get nutritional analysis to make sure that they have the vitamins and minerals that we want to target.
And you don't only want astronauts to eat microgreens, right?
There are so many other crops that we're researching here that we would love to see our astronauts take part in.
Is the goal to send crops to another planet?
It's ultimately our greatest goal, to have a full greenhouse full of plants, full of a wide variety of textures and flavors.
The work that you do is literally interplanetary. What does it mean for you to be a part of the team?
That I have left a valuable mark on science. If I stop tomorrow, the things that I've done up until now will have a lasting effect. It means that I've supported mankind's desire to go forth and explore. While we're not astronauts, we're not going to space, we're not riding rockets, but we are so important to the mission and the goal.
So as a woman and a woman of color, what does it mean for you to be opening up doors for other women to come behind you?
It means a lot to me. Me being here with my background is so important for people coming behind me, knowing that there's people here that have laid that trail for them.
Well, thank you so much, Lachelle.
So glad you came. Thank you.
I'm kind of in the mood for a salad. I don't know why, but I've got some good vinaigrette. Wanna go try some?
Sure.
All right.
Welcome back. Before we go, we have one last thing.
If you devote time to what you are passionate about, then you can help others using your passion.
My biggest advice is to not allow your first roadblocks, your first mistakes, your first closed doors to keep you from your goals. You can always rely on what you learned during those hardest times to get you through the next roadblock.
That's it for this episode of Mission Unstoppable. Next week, I'm trying to teach a turtle to take pictures of itself. We're calling them shelfies. See ya! If you're watching this, you must have really liked the video. Make sure you follow and subscribe, and check out these other videos that are even better. No, really. I've seen this one over 100 times.